Good afternoon. Welcome back to another lecture. So today we are going to be talking about topic 11, the infected cell. It is the Saturday of break, so it has been about a week and a half since classes let out. I hope that you all are doing well and that you all are staying safe. So hopefully between the last two videos and then this video, you're noticing an increase in the quality of the sound. So I actually went out and got a new microphone. I realized in the last two videos that I filmed, which happened to be the first two, so you all were the guinea pigs for me learning how to play with my setup and trying to get it to work and get it all figured out. But I learned in that old setup that there was a really weird echo and it was making me hate my voice even more than I normally hate my voice. So I got a new microphone, and I think that with the new microphone it cuts back on some of the echoes in the room. So hopefully it sounds better. Let me know if it does or if it doesn't. So before we get started, I just kind of wanted to share really quickly with you where we are at currently. So as of Saturday, March 28th at 5:03 p.m., according to the Johns Hopkins GIS website that is up, we are currently at 652,079 cases of coronavirus. The U.S. surpassed 100,000 cases yesterday, so we are currently at 116.5 thousand cases, and that is only rising, and I suspect that it will continue to rise. So if I go ahead and kind of zoom in on the US, you can see that they switched the map back over to looking at counties. So we can go ahead and zoom in on Indiana here. So in Indiana, just about every single county is now reporting at least one case. Of course, we're only reporting the people that have tested positive. A lot of people are being turned away just because they're not as ill as others. So just wanted to kind of touch base with you with regards to this and kind of show you where we are at. I hope that everyone is being safe. I hope that everyone is practicing social distancing and that we are all doing our part for humanity as a whole. So what we are going to do today is we're going to answer this overarching question of what happens to a cell once it is infected. And we're going to split this up into 4 parts. So we're going to talk about signal transduction, gene expression, metabolism, and remodeling of cellular organelles. So it's a little bit of a flashback, but if you all remember from topic 1 at the very beginning of the semester, we talked about this idea of a virus versus a virion. And so we said that a virion is an infectious particle, whereas a virus is the infected cell. So I wanted to remind you of this idea of virion versus virus. So we have spent the past most of the semester or so, I've lost track of how many weeks it's been with spring break and the world being turned upside down, but we spent the past couple of months mostly looking at things that happen from the virus perspective. So we have talked about how does a virus attach? How does a virus take its nucleic acid and copy it? How does it then translate that? How are all of the pieces of the progeny virions built together, and how does a virus exit? But what we haven't really done is we haven't really spent a lot of time talking about things from the perspective of the infected cell. So what we're going to do today is we're going to take an integrated description of the impact of a virus infection on a cell. So what I want you all to do is to first kind of just pause, and based on what we've talked about so far this semester, perhaps based on what you have heard in the news about a current coronavirus outbreak, go ahead and brainstorm your ideas and brainstorm what you think happens to a cell when it is infected. All right, so hopefully you've come back with lots of really great ideas. I will jot down just a couple, and I'm going to work off of what last year's class said, since you all aren't in the room with me and you can't tell me. And even though the dog is sleeping behind me, she has nothing to offer. So I guess maybe we can add that when a cell gets infected, it gets very tired. So in all seriousness, though, one of the main things that probably you all came up with is this idea of death. So when we talked about a virus exiting, we said that oftentimes apoptosis occurs. Of course, we can have budding, but even if we have budding that occurs, imagine the innards of that cell. They're completely and utterly annihilated from the virus. So even if the virus exiting the cell doesn't kill the cell will eventually, using cell signaling, will realize that it probably needs to undergo apoptosis. So regardless, typically when a cell is infected with a virus, it will result in death. Hopefully, you came up with something that has to do with energy production. So we know that nothing is free. We need energy to be able to do all of these pieces. So to make more progeny viruses, to make their protein components, their nucleic acid components, their envelope components, we need energy for all of that. And so the cell is of course going to be hijacked, and the virus co-opts the cell into tricking it to increase energy production for the virus's own use. And other things that we kind of mentioned is this idea of signal transduction. So there will be changes in signal pathways, and we'll talk a little bit about those today. And as a whole, we could take all of this and any of the other ideas that you all have, and we can say that normal cell processes stop or change, right? So when a virus takes over, that cell is no longer normal. A lot of things are going to be changing. And all of the things that are on this page and all the things that you all came up with, and if there's things that are not listed on here that you came up with, go ahead and put them in the comments in the video, because I want to see what other things you all came up with. But all of these things can be categorized into one of 4 categories. The first category is change in signal transduction, which is also known as cell signaling. The second change that we're going to have is change in gene expression. And we've touched on parts of change in gene expression. So when we talked about the control of viral transcription, we actually started to think about how viruses affect transcriptional control in hosts. So we'll come back to that today. We are also going to have metabolic changes. And so again, that deals with that energy production. And then last but not least, we'll also see that organelles are remodeled. So what we are going to do next is we are going to walk through each of these categories. And this entire topic, so topic 11, could probably be an entire semester-long course in and of itself. So we're going to kind of hit the highlights of each of them. And I'm going to have you all pull in information from this class and from other classes. And we're going to be building on prior knowledge. So let's go ahead and let's start with signal transduction. So cell signaling. Why is cell signaling important for a cell? And if you have had cell biology, the running joke in that class is it's important for everything. And it kind of is. So I'm going to go ahead and write important for everything. So my cell students used to tell me that if you didn't know what the answer was to a question in cell biology, you could probably get away with saying it had something to do with cell signaling. And while that is true, we're going to be a little bit more specific. So when we say that cell signaling is important for everything, you can probably come up with the idea of communication. So we have cell-to-cell communication that has to occur. One of the things that happens when a cell gets infected with a virus, it actually turns on through cell signaling a certain signal transduction pathway that releases into the extracellular space a molecule that lets the cells around it know, hey, I've been infected, start preparing, start to protect yourselves. But of course, viruses have ways to block that signal transduction pathway. But communication in a normal day-to-day life of a cell is something that is very important. Other things that cells have to do is that cells must sense the environment, and that refers to both internal and external, and they must respond to it. So it's not good enough to just sense it, but you have to be able to somehow respond to it. There's a lot of other ways that signal transduction is important for a cell. Basically, all of immunology is all signal transduction. When a cell determines whether to proliferate, to not proliferate, that comes from the idea of sensing the environment. That is all through signal transduction pathways. So we can summarize this by saying that signal transduction is going to govern every aspect of cell physiology. And you can imagine, if we are talking about this in a virology class, that viruses in turn must affect these pathways. And that is, in fact, very true. So viruses are going to change signaling in an effort to promote replication. So we're going to talk about some of the ways that viruses do this. So let's start with a signaling pathway. So this signaling pathway is the PI3K ACT mTOR signaling pathway. If you take a look at it, it looks like a walk in the park, right? Piece of cake, no big deal, super simple. Totally kidding, obviously. This is a rather complicated pathway, and we will simplify it here in a little bit, but let me tell you why this pathway is important. This pathway is one of the pathways that helps to regulate the cell cycle. So if you think back to Bio334, genetics, if you've taken cell biology, we spend a lot of time talking about regulation of the cell cycle. So this idea of growth, synthesis of DNA, a second growth period, and then going through mitosis, and then you end up with 2 daughter cells. So there are many signaling pathways that help regulate and control the cell cycle. The PI3K ACT mTOR signaling pathway, which I'm just going to call the mTOR signaling pathway just for simplicity, is one of the pathways that regulates the cell cycle. And so the entire goal of the signaling pathway is to sense the environment, and it's going to help a cell determine to live or not to live. And so it's going to help to determine, do we undergo apoptosis? Do we proliferate? Is it time to grow? We see the idea of autophagy on here. So we talked about autophagy as being important in exiting a cell, and so viruses can co-opt that as well. So let's kind of break down this pathway, and we'll walk through some of the pieces, and then on the next page we will actually draw a simplified version. So a couple of things that we need to know what they mean. The first thing is what an arrow means, and it drives me crazy that cell biologists and pretty much all biologists always do a green arrow and then a red to inhibit. And you'd think as biologists we would pick better colors knowing that people are red-green colorblind. But anyways, I digress. So if you have an arrow, regardless of the color that it is, it means that that pathway is stimulating or it is turning something on. If you have a red line with a straight line at the end of it, this means inhibit, or it means that it's turning something off. So if we take a look at the pathway over here on the left, you'll notice that we have a receptor tyrosine kinase here. Integrin is a protein that we have seen. GPCR stands for G-coupled protein receptor. So these are all of the proteins that are helping to sense the environment. You can see that they're turning on this PI3K. PI3K can turn on other things, and we're going to talk about kind of some of the proteins that you've heard of and some of the more important pieces. So here we see actin remodeling. So actin is part of the cytoskeleton. This is going to play a really important role, and I'll save the why here for the next few pages. You might notice cyclin. So cyclins play a role in controlling the cell cycle. Again, in cell biology, we spend a lot of time talking about cyclins. So if you are taking it next fall, we will spend some time on that. If you have taken it already, you should hopefully cyclins ring a bell. You may have also heard of P53 before. So P53 is going to help with DNA repair. These proteins BCL2 and BAX are actually important for apoptosis. Again, something that we discuss in cell biology. We have a whole nother arm of the pathway down here. You'll notice EIF4E. This is an initiation factor for translation. So again, lots of proteins, lots of pieces. You can see that many stimulate, many inhibit. And depending on what is on, what is off, it's going to determine how a cell reacts. So this is a really complicated pathway to look at. We're going to draw it simplified, and the simplified version is what you need for this class. So let's go ahead, and on the left here, we'll draw our simple model. So here's our plasma membrane. We're going to draw a little receptor. And as you all know, I envision receptors as either half circles or triangles or squares, depending on the mood that I'm in. And so this is the part of sensing the environment. And we are going to have some sort of a ligand or a signal. When that binds the receptor, that's going to cause a change. And I'm going to put this giant circle, and I'm going to write super scientifically, stuff happens. So when I say stuff happens, what this is really referring to is there's a lot of signal transduction that happens. It's kind of like when you play phone and you say, to the person next to you, hey, so-and-so said this, and the next person passes on the signal. So that stuff happens is all of the passing on of this signal. And depending on which proteins are turned on and which proteins are turned off, which in turn goes back to the idea of what ligand is binding, what is the input signal that we are sensing, we can get a couple things that can happen. We can turn on apoptosis, we can turn off apoptosis, we can turn on or turn off autophagy, we can turn on or turn off cell growth, or we can turn on or turn off proliferation. So these are going to be the main ones that we are going to talk about. So what I'm going to have you do is go ahead and pause the video, and based on what we have said so far, see if you can come up with ways that viral proteins might work on this pathway. All right, so hopefully you've come up with some predictions. If you are stuck, I'm going to give you a little bit of a hint. So I'm going to challenge you all to think about a virus, so an infected cell, from an early infection compared to a late infection. So let's draw and write out what I mean by that. So what if we have an early infection? So the virion has just infected the cell, If we think about from the virus perspective of what we need versus what we don't need happening, we can kind of start to hypothesize what might happen. So early on in infection, we probably don't want to turn anything on that has to do with exit. So apoptosis and autophagy are both ways that viruses can exit a cell. So early on in infection, the virus isn't thinking about that. So we're going to turn off apoptosis, and we're going to turn off autophagy. Okay, so that's going to be early on in infection. So as we continue through the infection cycle, we're going to be copying the nucleic acid. We're going to be making all of the components. Eventually, we're going to reach late infection. In late infection, this is where we are going to want to turn on either apoptosis, so we'll have an arrow, and turn on autophagy. Now, we can actually add to this, so let me go ahead and move this down. Oop. So other things that we can add are, well, what about growth? What about proliferation? When we first enter as a virus, we probably want the cell to grow because, well, it's going to take a lot of energy to make those progeny virions. So we're probably going to have on for proliferation and on for growth, and we'll have the opposite for late infection. We're probably going to have off for proliferation and off for growth. All right, so that's kind of takes care of this idea of early versus late infection. Let's go on to a new page and let's kind of talk about or think about lytic versus latent infections. So really great examples of latent infections are HIV, are herpes, and so for these types of pathways, for the lytic versus the latent reproductive life cycles, if you will, which ones do you think are going to not want to turn on apoptosis or autophagy? Probably the latent ones, right? So when we have our herpes viruses and our HIV, they're probably going to spend time turning on proliferation, right? The cells that are housing their genetic information, we don't want those cells to die. We want them to continue growing. Whereas we're going to turn off apoptosis and autophagy. For the lytic life cycle, this is going to be split up into the early infection and into late infection. Okay, so hopefully you all are still with me and I haven't lost you. So go ahead and in the comments below, let me know if there are other things that you came up with when you were working on this practice problem. So we're going to go ahead and we're going to do another set of practice problems. I'm going to go ahead and give you some time to work on this, although in real world, when you're watching it, it'll seem instantaneous, but I will see you on the flip side after you have had time to practice. Okay, so hopefully you have had some time to work through some of these problems. So let's talk a little bit about number one. So number one states that a virus has a mutation in a gene that inhibits its ability to turn off apoptosis. So you can imagine if a virus has a protein whose job is normally to turn off apoptosis and early infection, which means that the cell is probably perceiving there's something wrong, but it can't undergo apoptosis, this would likely be beneficial to the cell. So we may have, in this case, a cell that is able to undergo apoptosis, and we may result in no viral progeny or a decrease in viral progeny. Of course, it will kind of depend on exactly when this protein that is encoded by this gene is acting. And so for this one, we can argue that this may be beneficial to the host cell. So perhaps a decrease in viral progeny, or the infection is halted. Again, you could kind of make arguments for a multitude of answers. So these are just some of the explanations that I have come up with. So let me know in the comments below if you have other explanations for any of these three practice problems. So number 2, a virus has a mutation in a gene that inhibits its ability to turn on proliferation. So this is going to kind of depend on the type of virus. So let's say that we have a DNA virus. So we know that DNA viruses go to the nucleus. And when we talked about replication of DNA viruses, we talked about how they require the host to be in the S, or the synthesis part, because otherwise their DNA isn't going to get replicated. And so for a DNA virus, this could be completely horrible. This might result in no DNA replication. And if we don't have any DNA replication, that maybe gives the cell time to recognize that it is infected by a virus, and it gives it time to undergo apoptosis. Now, for an RNA virus, this might not have as bad of repercussions. Maybe we just have a decrease in viral progeny, right? You could make different arguments for the different types of viruses that we have. So again, these are just practice application questions to get you all thinking about potential test application questions. All right, so number 3, a drug, so an antiviral that interacts, VP stands for viral protein. So a drug that interacts with the viral protein that turns on autophagy. So what we normally have is a viral protein that turns on autophagy. Now we have a drug, and this drug is stopping that viral protein, which means we don't turn on autophagy. So if this happens, this might be a great way to stop a viral infection, particularly if those viruses rely on the autophagy pathway to get released into the extracellular space. So again, just wanted to give you all some examples of practice problems, and like I said earlier, let me know in the comments below if there are other things that you came up with, or if you have other explanations than the ones that I shared with you. So another thing that we can think about is, well, we have these viruses that are manipulating all of these different pathways. So if we go back for a second to our signaling pathway that we have viruses that are manipulating actin remodeling. So when viruses first enter, most of them are endocytosed in one way or another. Those are going to require actin to be remodeled. They're going to require actin to be remodeled as we use the cytoskeleton to move things in and out and to move things throughout the cell. They're going to affect cyclins, which are going to affect the cell cycle. They're affecting all of the multitudes of things that the signal transduction pathway controls. And so from there, we can hypothesize that there are absolutely going to be things that go wrong from the host side of things. So go ahead and see if you can come up with some ideas and some hypotheses of what could go wrong. All right, so hopefully you have come up with a couple of ideas, and I'm going to share with you the things that I have come up with. So all viruses have a protein, or one or more. They all have proteins in one fashion or another that alters something in a signal transduction pathway. Now, usually, this isn't the worst thing in the world because most viral infected cells die. But those that survive, you can imagine, are going to have permanent changes and permanent damage. And so one of the things that can happen is things like tumors, cancer. So for this, we're actually going to have an entire topic that is all about oncogenesis. So how do viruses lead to cancer? And some of the viruses that can do this and can affect the host are things like herpes viruses, human papilloma viruses, polyoma, and HIV. So I'm going to leave you with that for today because, like I said, we are going to have an entire topic on just this idea. But this is something to keep in mind, is that even though most of the time it's not an issue because the host cell dies, when it doesn't, it can have huge repercussions on the host as a whole. All right, so that wraps up our signal transduction discussion. We're now going to move into gene expression. So again, we're going to start with a discussion question. So hopefully the things that I tell you are some of the things that you brainstormed. If they're not, if you come up with things that I don't have listed, be sure to comment them in the comments beneath so that your classmates can see. So when we think about how and where gene expression may be affected, it can be affected at any point of gene expression. So what that means is it can affect gene expression during transcription, during processing of our mRNA. So when we go from that pre-mRNA to our mature mRNA, it can affect it during export. It can affect it during translation. Any part where gene expression can normally be changed in the host, a virus can affect it. So in terms of how do viruses do this? They typically do this with viral proteins, although viral mRNAs can be involved as well. So let's break it down into some of the main pieces. So if we have transcription, we can have transcription factors that are degraded. So if you have no transcription factors, those don't have, you don't have anything to bind to your promoters and your enhancers, and so you turn off that gene expression. Other things that we can do is mRNA of host is degraded. So lots of different ways that viruses can do this. Some viruses actually encode for nucleases that their entire job is to recognize host mRNAs and chop them up. You can also have no production of mRNA, so of host mRNA, and we do this by changing the DNA to heterochromatin. So when we talked about control of gene expression for viruses, we also started to hint at how viruses affect hosts. So we talked about there are ways that viruses remove acetyl groups from those histone tails, which now means that instead of heterochromatin instead of euchromatin, that now we have heterochromatin, so that DNA is wound tightly. Now the transcription factors can't get in. You can also imagine that you can have activators and repressors that are destroyed or blocked. So I'm going to refer you back to that topic for some of the details of that. Other things that we can do. We can stop splicing. So we know that splicing is a really important part of processing pre-mRNA to mature mRNA. So splicing can be stopped. We can also stop viral RNA degradation. So just like viruses have nucleases, host cells also have a lot of endo and exonucleases that help determine the lifespan of an mRNA. And so some viruses encode for proteins that stop degradation, because if you have an endo or an exonuclease that chews up mRNAs, it's not going to discriminate between virus or host. And so there are viral proteins that stop that from happening. We can also stop export of mRNAs. So just like how HIV tricks the host into thinking that its genome has been spliced so that it can be exported into the cytoplasm to be packaged, there are proteins that can stop export, and so they block that signaling that affects this part of gene expression. So there's a lot of different ways, and these are kind of just the tip of the iceberg. So I'm going to go ahead and show you a picture here. And I really like this picture because I think it does a really nice job of summarizing everything that we just said. So we've seen this image before. We've actually talked about this process. So we've talked about how DNA, how we take that and we transcribe to RNA, how this pre-mRNA is then both polyadenylated, capped, and processed to make mature mRNA, how it is exported, and then we touched briefly on translation. So I don't expect you to know all of these proteins or all of these viruses, but this is to just reiterate the idea that gene expression can be affected at every and all steps. So poliovirus, VSV, and many alpha viruses have proteins that actually come in and they stop RNA polymerase II. So RNA pol II is the main polymerase. Some viruses also actually have proteins that will stop RNA pol 1 and 3, which are responsible for some of the other RNAs in the cell, but they get right into polymerase and they just stop it. And so now we don't have any host mRNAs that are made. Some viruses, like our enteroviruses and influenza, will actually mess with the machinery of going from pre-mRNA to mature mRNA. So HSV1 and enterovirus also does something very similar. So these viruses can affect the capping machinery, the splicing machinery, and the polyadenylation machinery. And we know that in order for a mature mRNA to exit the nucleus, all three of those things must be completed. And so by stopping and interfering with one of those steps, we stop that process from happening. We also here have poliovirus, VSV, and then we've got adenovirus over here. So these viruses also affect export, so they can stop that process from occurring. And again, this is all with a goal to decrease the amount of host mRNA to make the virus mRNA, if they make mRNA, and to control gene expression in favor of making more virions and making more progeny virions. Over here, we also have degradation. So mRNAs are normally degraded. They have a certain lifespan. Some are shorter, some are longer. It's one of the ways that the host actually controls gene expression. And one of the ways that some viruses, including coronavirus, so here is coronavirus, and it uses a protein NSP1, so it's a non-structural protein 1. super specific name there. But what these coronaviruses do is they actually degrade the host mRNAs. And that allows for the coronavirus RNA to be the more populated one in the cytoplasm. So again, this is to just show you an overview of how and where gene expression may be affected by viral proteins. So, okay, this kind of makes sense for DNA viruses, right? And we saw some RNA viruses on there, and so clearly RNA viruses do this as well. But why? Why do RNA viruses want to interfere with transcription? Most of them don't even go to the nucleus. So the question is, why do they care? So go ahead and see if you can come up with an answer. All right, so what'd you come up with? Did you figure it out? If you think back to one of the first topics that we talked about in this class, we talked about that every virus, doesn't matter what type of virus you are, must make mRNA. And not only does it make mRNA, it must make mRNA that the host ribosomes can read. So in this class, we've been talking a lot about unifying principles of all viruses. And this is one of those unifying principles. And our Baltimore scheme of classification that we've been looking at has mRNA right in the middle, and it organizes viruses based on how do they get to that mRNA? How do they make more of whatever their nucleic acid is? So every single virus, we have not discovered one yet that doesn't do this. Thus far, every single virus must make mRNA. And so the idea is that you eliminate the competition. So viruses are definitely super jerks. We want to get rid of anything else that could potentially be competing. So even if you are a positive sense RNA virus that is in the cytoplasm that never makes it to the nucleus, if there's a bunch of host genes that are being expressed, your mRNA, your RNA genome, is going to get lost in the cytoplasm with all of the other ones that the host is making. And so it makes sense, regardless of whether you're a DNA or an RNA virus, get rid of the competition. All right, so the last thing that we're going to cover in day 1 of topic 11 is translation. So we've mostly been focused on transcription. We did say that viruses can affect translation, but we didn't really talk about in details. And so the answer to this question is heck yes, absolutely. Viruses can affect everything in a cell. So there was a really cool experiment that was done with poliovirus, and what the researchers did was, over time, they looked to see, so over time, what they looked at was protein synthesis rate. So what they saw was, in an uninfected cell, protein synthesis goes up. So in an infected cell, however, we see protein synthesis goes down, and then it will go up and it'll decrease again. So when it goes down, this is host protein translation. So as viral infection continues over time, the host protein translation decreases, and then over here where we see this giant increase, this is viral protein translation. So as infection goes on, eventually it's time to start translating those viral mRNAs to make all of our pieces to assemble the progeny virions. So again, this was shown in poliovirus, but many viruses do this, and this applies to a lot of them. So now let's talk about how. How can viruses do this? How do they go ahead and stop this idea of translation? So just like we have different proteins that can affect transcription, we have different viral proteins that can affect translation. One of the ways that viruses affect translation is to break initiation factors. So one of the initiation factors that plays a very important role is EIF4E. And what this initiation factor does is it recognizes the cap on our mRNA and it directs the ribosome towards it. So one of the things that viruses do is they actually have proteins that will break off the part of this initiation factor that recognizes the cap. So if that protein is broken, now it can't recognize the cap, and now you can't have translation of that mRNA. And so what viruses do instead is they actually have something called cap-independent translation initiation. So whereas eukaryotes have cap-dependent translation initiation, which is the whole role of EIF4E, viruses actually start translation in other places. So they will start it a little further into their mRNA. So again, viruses are affecting absolutely everything. And again, the goal here is to help ensure maximal viral gene expression. We don't want host gene expression, we want viral gene expression. So we are going to stop day 1 here, and when we come back for day 2, we're going to talk about how viruses affect metabolism and how they remodel cellular organelles. So thanks for hanging out with me, and I will see you really soon. Bye everyone.